Okay, we'll go ahead and get started. My name is Todd Herron, and I want to welcome you to Greenstone Bioscience's second webinar. The title of today's webinar is New Approach Methodology, NAMs, for Preclinical Cardiac Safety Screening. Today we'll be focusing on hiPSC-derived cardiomyocyte electrophysiology assays, and we'll review how these NAMs provide value beyond the traditional hERG ion channel assay. And the goal is to learn how human stem cell derived cardiac NAMs from Greenstone Biosciences can be used for preclinical efficacy and therapy safety assessment. So thank you very much for joining us today. Just to give you some orientation, we're broadcasting today from Greenstone Biosciences laboratories and offices, which are located in Palo Alto, California, at the Stanford Research Park. We invite you to follow us on Blue Sky, Twitter, or LinkedIn. And you can please check out our website at greenstonebio.com. So just to give a little background review on Greenstone Biosciences Incorporated, we're a biotechnology startup company located in Palo Alto, California. Our founders are Jade Chow and Professor Joseph Wu from Stanford University. Our company, Greenstone, is rooted in 2 decades of new approach methodology development using iPSCs as NAMs from Dr. Wu's laboratory at Stanford. We also have a drug discovery pipeline and have 2 different molecules that are in clinical trials. And one of our main strengths is our iPSC Biobank, which contains over 2,000 ready-to-use patient cell lines that can be thawed and mobilized to create human new approach methods for a variety of different uses. This is an example of some of the publications from Dr. Wu's laboratory that Greenstone is founded on. Dr. Wu has published with some leaders, Nobel laureates such as Dr. Yamanaka in 2016, providing perspective on how iPSCs have progressed over the years to become meaningful new approach methodologies for use in drug discovery and drug development projects. Dr. Wu has also published many other papers in very high impact journals over the years. Here you can see one from Cell Stem Cell in 2022, review of the concept of clinical trials in a dish for cardiovascular medicine in 2024, and reviews on drug discovery and the use of iPSCs in 2024. And more recently, in the journal Science, Dr. Wu's laboratory published a report on using gastruloids and show how they enable modeling of the earliest stages of human cardiac and hepatic vascularization. To give you some introduction into my background, my name is Todd Herron. I'm a PhD scientist by training in molecular and cellular cardiology with focus on use of pluripotent stem cells. I completed postdoctoral training at King's College London and also at the University of Michigan. I have over 15 years of experience in the hiPSC field, developing new approach methods, specifically for cardiac safety screening. I served as a faculty member at the University of Michigan in the Internal Medicine Department in the Center for Arrhythmia Research for 20 years, and recently founded the University of Michigan Cardiovascular Regeneration Corps. My current position, I've moved to Greenstone Biosciences and am the director of the iPSC Biobank. And I also am a proud member of the HESI Organized Cardiac Safety Committee, a critical member of the iPSC-Cardiomyocyte validation team since 2016. And I'm excited to share how this committee's work has validated human cardiac NAMs and how this is changing the regulatory science and regulatory filings at the FDA. So this is our second webinar. In our first webinar, in case you missed it, you can find it on YouTube, or you can also find it on our website. The first webinar focused on NAMs in general and focused on how the FDA and the NIH are promoting the use of NAMs for drug discovery projects. And the title of that webinar was Bringing Human Biology to the Earliest Stages of Drug Discovery Programs Using Patient iPSCs. In that webinar, I introduced Greenstone Bioscience's custom differentiation services and reviewed in general the broad variety of cell types that we can provide for use as new approach methodologies for drug discovery projects. This is a review of those cell types on the top portion of this slide and 3D organoids that are being used and developed at the bottom of the slide. Today's webinar is going to focus on cardiovascular NAMs. So I'll be focusing on primarily cardiomyocyte assays, and also we'll introduce some three-dimensional cardiac organoid products that we've developed here at Greenstone Biosciences that are available for drug discovery and cardiac safety screening. So for our second webinar today, this is the agenda of topics that will be covered. I'll briefly review the traditional preclinical cardiac safety screening approaches and paradigm that focus on the use of the hERG ion channel assay and the QT interval of the electrocardiogram. I'll introduce human-induced pluripotent stem cell-derived cardiomyocyte assays and the validation that's been done of this new approach methodology by international multi-site validation studies. Importantly, now iPSC-Cardiomyocyte assays have started to gain acceptance in preclinical safety assessments for IND applications to the FDA. And I'll review at the end of the webinar some of the Greenstone Biosciences iPSC cardiomyocyte assays that are available for your drug discovery projects and for drug development projects. So the primary goal of preclinical cardiac safety is to detect a compound or other therapeutics risk to alter the EKG or the ECG. And this focuses on any changes of the QT interval that might cause arrhythmias in patients. So what you see on the bottom left is an EKG trace, which has a QRS complex and then a T wave following at the end. And this interval between the Q and the T points of the EKG, called the QT interval, is what's really focused on in preclinical cardiac safety screening. And what you see on the right is in preclinical cardiac safety, the goal is to try to detect a compound or an antibody therapeutic or any kind of gene therapy and its effect on the QT interval. So in this example on the right, the red trace shows the effect of a drug that prolongs the QT interval, which you can see at the bottom, and also causes a reentrant arrhythmia known as torsade de pointe. So this is the effect of the EKG that preclinical cardiac safety screening is really trying to prevent any of this being released to patients that can cause untoward side effects and can be dangerous for patients. Here I'm going to review the cellular and molecular basis of the current paradigm of drug-induced QT prolongation and arrhythmia. The screening at the moment focuses initially on the effect of a drug to block the hERG ion channel. hERG stands for the human ether agogo related gene. This is a gene that encodes an ion channel that is responsible for potassium ion channel conductance across the membrane and is one of the primary determinants of repolarization of the action potential, which is what you see the trace in the right of the single cardiomyocyte that's being patch clamped. So in black, the black trace of the action potential shows a normal baseline action potential, and any drug that blocks the hERG ion channel, the effect on the action potential is shown in red, where the action potential duration becomes prolonged, and by inference, This is thought to also contribute to the QT prolongation that's observed in patients on the EKG assessment. So by inference, any compound or new therapy that is found to block the hERG ion channel is assumed to cause QT prolongation of the EKG and increase the risk for development of a fatal ventricular arrhythmia called torsade de pointe. So APD prolongation in vitro is assumed to also be associated with any QT prolongation that might be experienced by a patient clinically taking a drug such as the one shown here. The traditional preclinical cardiac safety screening approaches focused on the hERG ion channel. The hERG ion channel block is used as a surrogate in vitro marker for action potential, duration, and QT prolongation. And at the moment, in drug discovery projects, any therapeutic compounds or other modality of therapy found to block the hERG ion channel are typically discontinued from further development due to fear of cardiac liability risk where the compound may cause arrhythmias in patients. This assay has been found to be potentially too sensitive of an assay and does produce false positive signals where compounds that are actually safe are mislabeled as being a cardiac liability that may cause arrhythmias in patients. So the current paradigm is shown in the cartoon at the bottom, where patch clamp experiments focused on the hERG ion channel are tested. Drugs are given in this assay. Any drug known or found to cause blockade of the hERG ion channel is assumed to cause action potential duration and lead to QT prolongation and risk for development of torsade de pointe that can degenerate to ventricular fibrillation, a fatal cardiac arrhythmia. The current use of the hERG ion channel is shown here in this slide. So typically genetically modified human embryonic kidney cells, HEK293 cells, or CHO cells are used in this assay. And in this assay, these cells are genetically modified to overexpress the hERG ion channel, as you can see in the bottom. And then these immortalized cell lines are used for cardiac safety screening and hERG channel ion blockade assessment. And at the bottom, you can see any drug found to inhibit potassium current through the hERG ion channel is presumed to lead to QT interval prolongation and increase the risk for development of a drug-induced arrhythmia. However, this might be an oversimplification of a complex biological problem. So one limitation of the hERG channel cardiac safety assay that's used predominantly in preclinical safety screening is that human embryonic kidney cells do not recapitulate the complex ion channel expression of human cardiomyocytes. The human cardiomyocyte, besides expressing the hERG ion channel, also expresses sodium channels, calcium channels, and other potassium channels that also repolarize the action potential of the cardiac tissue. And these other ion channels, if a drug blocks the hERG ion channel, these other ion channels often can compensate for that hERG block to maintain a normal rhythm of the cardiomyocytes. So you can see the difference in this slide between the currently used HEK293 hERG assay shown in the top, where a single ion channel is tested for cardiac safety. On the other hand, in the human cardiomyocytes, there's a full complement of ion channels that are expressed, including calcium, sodium, and potassium ion channels that work together to form the action potential of every heartbeat. Another limitation of the hERG channel cardiac safety assay is that not all compounds that block the hERG channel actually cause arrhythmias. For example, the compound fluoxetine, which is known as Prozac, blocks the hERG ion channel. However, even though fluoxetine blocks the hERG ion channel, it has been a widely used medication and is considered to be a safe medication with very little cardiac arrhythmia side effects. Fluoxetine was developed before the hERG assay became the gatekeeper of drug discovery funnels. If fluoxetine were to be discovered now, it may represent a potentially missed opportunity because fluoxetine would be labeled as a therapy that carries a risk for development of cardiac arrhythmias. About a decade ago, a movement in the field called the Comprehensive In Vitro Proarrhythmia Assay, or CIPA for short, promoted the use of human iPSC-derived cardiomyocytes to complement or to replace the hERG assay, as it was recognized that the hERG assay may result in potentially missed opportunities where drugs and other therapies that are actually safe are mislabeled as being cardiotoxic and carrying a risk for the development of fatal cardiac arrhythmias. So the CIPA committee has promoted the use of human-induced pluripotent stem cell-derived cardiomyocyte assays as a new approach method, or a NAM, that may complement or may replace the hERG channel safety screening assay. hiPSCs are derived from patient blood or other somatic cell source, could be skin or hair. The PBMCs can be readily and very efficiently now reprogrammed to human-induced pluripotent stem cells, which are very similar to embryonic stem cells in the way that they are cultured and the way that they divide in culture. The iPSCs represent a cell type that self-renews continuously. And so once hiPSCs are generated, they represent a virtually unlimited supply of patient cells that can be used for in vitro screening. iPSCs can be readily and with very high efficiency now differentiated to functional cardiomyocytes. And remarkably, these cardiomyocytes can be cryopreserved for distribution to laboratories across the world. These cardiomyocytes, when thawed and plated into high throughput screening plates, will form monolayers of cells that are electrically and mechanically connected that are very useful for preclinical cardiotoxicity and proarrhythmia detection screening. In 2018, an international multi-site study of human-induced pluripotent stem cell-derived cardiomyocytes for drug proarrhythmic potential assessment was published in collaboration between the FDA laboratories and the HESI committee, and validated the use of iPSC cardiomyocytes and showed their value in predicting compounds with known risk to cause cardiac arrhythmias. In this publication from FDA laboratories and the HESI committee, you can see on the left is a summary of the findings. So for in vitro cardiac electrophysiology models, human-induced pluripotent stem cell-derived cardiomyocytes were used in different platforms, including microelectrode array recording systems, as well as in voltage-sensitive optical recording systems. In this multi-site international validation effort, all laboratories followed one consensus protocol, using standard human-derived cell lines. Laboratories participated from 3 different continents and tested 28 different medications with clinical known risk to cause cardiac arrhythmias. Statistical and regression models were made, and the outcome of the assay and the validation of the cardiomyocytes showed an 87% pro-arrhythmia risk prediction score, which is very good for a predictive assay to detect cardiotoxicity and pro-arrhythmia potential of compounds. This validation effort led to iPSC-Cardiomyocyte best practices guidelines and publications. And now, after about 12 years of data collection, it's very exciting that the FDA now accepts iPSC-Cardiomyocyte data for IND applications. And an example of that is a publication from Toxicological Sciences from the FDA, which provided a contemporary review on the current use of human iPSC cardiomyocytes in non-clinical regulatory studies for cardiac safety assessment. So in this publication from the FDA and the HESI committee, 3 different approaches were tested, 3 different methods of recording the electrical activity and the functional activity of iPSC cardiomyocytes were validated. One in panel A is transmembrane recordings, which may be microelectrode recordings or optical recordings utilizing voltage-sensitive dyes. And here you can see, in response to the very specific hERG ion channel blocker E4031, you can see for voltage sensing dyes that the prolongation of the action potential is very easily measured and recorded in response to hERG channel blockade. In panel B, you can see the international validation study efforts also utilize data recorded from MEA or microelectrode assays, microelectrode arrays using field potentials. And here is an example of a drug known to prolong the QT interval, cisapride, and using multi-electrode arrays, one can also detect changes of the repolarization phase of the cardiac cycle and can infer a drug's risk to cause any kind of cardiac arrhythmia. Finally, in panel C, also contractility and motion based assays were validated. Here you can see for contraction measured by quantification of the calcium transient of each heartbeat. You can see in response to dofetilide, which is a very specific hERG ion channel blocker, very dramatic and very easy to detect prolongation of the calcium transient in response to treatment with dofetilide. And you can see, besides calcium transient measurements, also systems that utilize impedance to measure contraction, that one can also detect hERG channel blockade in response to E4031. In 2020, in Regulatory Toxicology and Pharmacology Journal, the best practices guide for using iPSC cardiomyocytes in preclinical screening pro-arrhythmia assays pointed out that the highest granularity for electrophysiological function comes from transmembrane recordings or optical recordings utilizing voltage-sensitive dyes to directly measure the action potential of iPSC cardiomyocytes. At Greenstone Biosciences, we utilize voltage-sensitive dyes and calcium-sensitive probes in high-throughput screening assays as a new approach method to screen molecules for preclinical cardiac safety. We utilize a high throughput electrophysiology plate reader called CARTOX. that measures action potentials utilizing voltage-sensitive dyes, or calcium transients utilizing fluorophores or genetically encoded calcium indicators. In the movie playing on the right, here you can see a 96 well plate of iPSC-Cardiomyocytes. Each well of this 96 well plate contains monolayers of cardiomyocytes, roughly 75,000 cells. Purified cardiomyocytes are plated in each well so that a confluent monolayer forms. In this monolayer, the cardiomyocytes are electrically connected and mechanically connected, and you can see very nice propagation of electrical activation waves traveling through each monolayer with each spontaneous contraction. On the bottom left, you can see the readout that we obtain from our high throughput electrophysiology screening assay. And we also have software for automated peak detection to quantify the depolarization phase, which is shown by the upstroke of the action potential in green, as well as the repolarization phase of the cardiac action potential shown in red. And this allows us very good granularity to determine the effects of any compounds effect on hERG ion channels, sodium channels, and calcium channels. This cardiac NAM, the cardiac electrophysiology NAM utilizing iPSC cardiomyocyte monolayers, has been validated. Here you can see on the top right a 30-second recording of spontaneously contracting iPSC cardiac myocyte monolayers, and you can see very stable action potential recording over this 30 seconds. And on the bottom is a zoomed in examination of a 10-second recording of spontaneous action potentials showing the normal rhythm that is characteristic of these cardiac monolayers at baseline. Using this high throughput electrophysiology plate reader, we can acquire data very rapidly. The machine is automated and can collect data from all 96 wells in about a 5-minute period. The assay enables detection of sodium channel block, calcium channel block, as well as potassium channel block. The system is useful for recording spontaneous action potentials, as well as calcium transients, which can serve as a surrogate marker for contractility of the cardiomyocyte monolayers. Now I'm going to go through a few validations that have been performed using iPSC cardiomyocytes. And this example shows how the cardiomyocytes can be used to detect calcium channel block. In this case, nitrendapine, which is a clinically used calcium channel blocker, was applied to iPSC monolayers at 4 different doses. The dosing was recommended by the hERG-CIPA committee as well as FDA safety regulators, and the doses are based on the Cmax that is recorded and known to occur in patients receiving effective doses of these medications. So the doses, there are 2 doses near the Cmax and 2 doses, the higher doses, are well above the Cmax reported in patients. And here you can see that for this calcium channel blocker, we can actually detect APD shortening, which is opposite of what you would expect to see if a molecule blocks the hERG ion channel. Nitrendapine is considered low risk to cause arrhythmias, and here you can see even at the highest dose of nitrendapine that the cardiac monolayers continue beating with a very normal and very rhythmic beating profile. Here's another validation of this iPSC monolayer cardiac new approach method, validated for detection of potassium channel hERG blockade or hERG block detection. This particular compound is called domperidone. This reagent is a dopamine antagonist and is known to have an intermediate risk to cause cardiac arrhythmias. And as you can see, with escalating dose going from 0.03 micromolar up to zero point 0.3 micromolar, you can see action potential duration prolongation. And at the very highest dose, we actually can see the development of drug-induced arrhythmia. So here, the activation at 3 micromolar, you can see that there's a repolarization delay, and the action potential actually doesn't come back to baseline, but rather stays in an activated state with very rapid firing in that torsade de pointe-like arrhythmia formation. So this is one of the real strengths also of the iPSC cardiomyocytes, is it's a movement beyond just ion channel blockade to actual detection of cardiac arrhythmias in vitro. Here's another example, another validation experiment using a compound that's known to have chemotherapy-induced cardiotoxicity. And this example shows testing of a drug called vandetanib, which is a tyrosine kinase inhibitor utilized as an anti-cancer therapy with well-known high risk to cause arrhythmias and action potential duration prolongation. And as you can see from the action potential traces shown in blue, that with escalating dose, escalating amount of vandetanib applied to iPSC cardiac monolayers, that we can start to see the development of arrhythmias, which you can see at the 1 micromolar dose, where there's rapid firing of 3 activations before a relaxation of the membrane potential. And at the very high dose of 10 micromolar, we can see extreme prolongation of the action potential duration and also the development of early after depolarizations, which are known to be a mechanism that lead to fatal arrhythmias, fatal ventricular arrhythmias, the torsade de pointe type of arrhythmia. Here's another example of a compound that's also known to block the hERG ion channel, the potassium current of the hERG ion channel. This drug is called di-sotalol and also is known to have a high clinical risk to cause fatal arrhythmias. Here again in this example, you can see with escalating dose up to 100 micromolar, very pronounced prolongation of the action potential duration, which indicates hERG channel blockade. Finally, we also have validated this cardiac electrophysiology new approach method using sodium channel blockers. Because we're using monolayers that conduct electrical signals and propagate waves of action potentials, we can generate activation maps for these cardiac monolayers, and we can measure the conduction velocity of the action potential or the calcium wave as it propagates through the electrically and mechanically connected cardiac myocytes. On the right, you can see for 3 different sodium channel blockers, for mexiletine on the top, disopyramide in the middle, and quinidine at the bottom. You can see that with increasing dose of each compound, that the conduction velocity, which is plotted on the y-axis, decreases as the dose of each of these compounds increases. Sodium channel blockers are well known and are actually used to slow the conduction velocity of the cardiac action potential. And here you can see that we can indeed detect mechanisms of sodium channel block in iPSC cardiomyocytes. So far I've provided a good comparison between the traditional hERG assay that is utilized and considered to be the gatekeeper for drug discovery projects, where it's a very simplified in vitro system using non-cardiomyocyte cells, actually using human embryonic kidney cells that are genetically modified, and then utilized in high throughput patch clamp assays to determine a drug's effect to inhibit the hERG ion channel. On the other hand, more modern NAMs for cardiac safety screening and pro-arrhythmia assessment utilizing human iPSC-Cardiomyocytes, where the cardiomyocytes contain and express the full complement of cardiac ion channels, including calcium channels, sodium channels, and other potassium channels besides the hERG ion channel, with very quantitative and rigorous and reproducible quantification of the action potential of the cardiac myocyte and the cardiac monolayers to get a more granular examination of the effect of a drug compound on the human cardiac action potential and electrophysiology. So Greenstone Biosciences, as I alluded to at the beginning of the webinar, we have a very large biobank containing over 2,000 patients' iPSC lines that can be utilized and mobilized for accelerated drug discovery and development programs. On the right, you can see the breakdown of our iPSC biobank in the pie chart. The bulk of our iPSC lines are for patients with cardiovascular disease, which represent 35.4% of the total biobank. So these patient cell lines, which are derived from patients with predominantly inherited cases of cardiomyopathy or arrhythmias, lend themselves to the development of cardiovascular disease and a dish models that can be very useful for cardiac safety screening, as well as efficacy screening for drugs in development to treat cardiovascular diseases. This table represents a glimpse of some of the iPSC lines and cardiovascular diseases that are available, as well as the genetic variants for each of those diseases. And I want to highlight for dilated cardiomyopathy, or DCM, caused by LMNA mutations, which are a rare mutation that cause dilated cardiomyopathy. And our iPSC biobank actually contains 36 unique LMNA DCM patient iPSC lines that can be mobilized and utilized for testing any compounds effect against a population of patients' cardiomyocytes that are more susceptible to the development of drug-induced arrhythmias. This also represents a very promising approach for efficacy screening of development of new drugs, where a compound that is meant to treat dilated cardiomyopathy can be tested against 36 unique patient cell lines, where each patient has a mutation in the same gene. So utilizing our biobank at Greenstone Biosciences, one new approach methodology that is very exciting is known as clinical trial in a dish. And in this paradigm, in this new approach methodology, if you partner with Greenstone Biosciences, a customer can select the patient population, which could be healthy control patients or could be disease patient cells for any disease. These cells from these patients can be thawed and expanded in our laboratories at Greenstone and differentiated to cardiomyocytes that can then be used in 2D monolayer assays and high throughput screening plates, or can be used to generate three-dimensional organoids, which are an expanding and rapidly developing new approach methodology being used in preclinical drug discovery. The patient population here can be customized by demographics of patients, can be customized by disease. So one could study just HCM patients in this paradigm, or just DCM patients, or the patient selection can be done by gene mutation specific cohorts of patients where the same gene is mutated but in different sites that lead to the same disease. And finally, there are many rare diseases represented in our biobank. Rare diseases such as arrhythmogenic right ventricular cardiomyopathy, or ARVC, also are a very useful new approach methodology and provide a cardiac substrate that has enhanced sensitivity to drug-induced cardiotoxicity. This movie is an example that I'll play of a monolayer of cells generated from iPSCs from an ARVC patient, and then the response of this ARVC patient's cardiomyocytes to hERG channel blockade. So you'll see on the left is baseline. You can see there's a pacemaker site, and we can see very good unidirectional conduction of the cardiac action potential from left to right. And at the bottom, you can see normal rhythm at baseline for these cardiomyocytes. However, when we add the hERG channel blocker E4031, we can see that this normal unidirectional activation pattern observed on the right actually turns into a reentrant arrhythmia, which is characterized and reminiscent of what occurs in the heart during ventricular fibrillation during a fatal arrhythmia known as TDP, or torsade de pointe. And here you can see reentrant activation pattern characteristic of those fatal ventricular arrhythmias. So here again, this is a good example of how the iPSC-Cardiomyocyte new approach method goes beyond just determination of hERG channel block, but actually can reveal mechanisms of cardiac arrhythmias. Moving from ARVC, here I'm providing some examples of dilated cardiomyopathy in a dish, showing that we can detect a hypocontractile phenotype for patients, in this case with an FLNC gene mutation. So here on the left, you can see characterization and comparison of DCM iPSC cardiomyocytes in the filled circles compared to a healthy control cell line. Here, the calcium transient amplitude was recorded and utilized as an index of contractility for each of the different cell types. And on the right, we can see that in response to hERG channel blockade, that the control patient cells continue to contract. However, the DCM patient cell line with this mutation did not tolerate the hERG channel blockade and actually stopped contracting at the same dose of E4031, indicating that DCM patient IPS cardiomyocytes do not survive hERG channel block and have an enhanced sensitivity to drug-induced cardiotoxicity and cardiotoxicity effects, which this adds real value to drug discovery projects to be able to determine a drug's impact on healthy cardiomyocytes and what one might expect from the EKG and the response, and how that compares to patients with more vulnerable cardiac substrates, such as patients with inherited DCM or inherited ARVC. Beyond the DCM contractile, hypocontractile phenotype, at the same time in this assay, recording the calcium transient amplitude as a marker of contractility. We also get a measure of the rhythmic activity, or the rhythm of the cardiac monolayers. In this case, healthy control compared to DCM in a dish, which you can see is also an arrhythmia in a dish. The DCM calcium transient analysis shows prolonged calcium transient duration, and also the occurrence of early after depolarizations, which are known to be a trigger and a cause for the development of fatal arrhythmias. On the bottom, you can see for the healthy control cohort or the healthy control patient wells of a 96 well dish, there were very little spontaneous arrhythmias that occurred in this population of iPSC cardiomyocytes from a healthy control patient. On the other hand, 13 out of 15 wells representing dilated cardiomyopathy in a dish had spontaneous arrhythmias. So this also shows that the iPSC cardiomyocytes represent an arrhythmia in a dish model that can be very useful for safety screening as well as for efficacy screening for the development of any new compounds to treat cardiovascular diseases such as dilated cardiomyopathy. So another way that companies or academic groups can partner with Greenstone Biosciences for use of iPSC NAMs. Also, we can utilize customer-provided iPSC lines. We can expand those iPSC lines to large numbers of iPSCs for bulk differentiation to cardiomyocytes that can then be used in two-dimensional monolayer assays or in three-dimensional organoid assays. Some new developments in the new approach methods for cardiovascular proarrhythmia screening and cardiotoxicity screening here at Greenstone involve the development of cardiac chamber specific cardiomyocytes and cardiac chamber specific organoids. So here you can see that for any patient, iPSCs, we can differentiate the iPSCs to either ventricular specific cardiomyocytes using established published protocols or to atrial specific cardiomyocytes. This chamber specific differentiation is important to recapitulate the function and the structure of the intact heart, where the intact heart is a four-chamber heart, where the atria are the upper chambers, which are low pressure chambers, and the bottom chambers comprise the ventricular chambers, which have a much different function and structure compared to the cardiomyocytes of the atrium. These cardiac myocytes can be mixed with other cell types, such as fibroblasts and endothelial cells, to generate organoids. We can generate chamber-specific ventricular organoids or atrial-specific cardiac organoids. And now we've had success and can provide these organoids as cryopreserved vials of organoids that are ready to use. And these organoids, as well as cardiomyocytes, have very high use potential for electrophysiology screening, for safety and efficacy screening, also for gene expression and detection of markers of disease or maturation, and for high throughput screening of drug responsiveness. For validation of cardiac chamber specific cardiomyocytes and organoids, using published protocols by Western blot analysis, you can see that atrial cardiomyocytes don't express beta-myosin heavy chain, but ventricular cardiomyocytes do express high amounts of beta-myosin heavy chain, which is quantified in panel B, where you can see atrial cardiomyocytes detect almost no levels of beta-myosin, whereas ventricular cardiomyocytes derived from iPSCs express very high levels of beta-myosin, and when we mix the two together, we get an intermediate expression. This myosin heavy chain expression profile mirrors what is known to occur in the intact, healthy human heart, where it's known that the atria express alpha myosin heavy chain, which is encoded by the MYH6 gene, while the ventricles express almost exclusively beta-myosin heavy chain, encoded by the MYH7 gene. And in panel C, you can see the distinct functional differences between atrial cardiomyocytes and ventricular cardiomyocytes, shown in blue, where atrial cardiomyocyte monolayers have a very short duration action potential relative to the ventricular action potential, which has much more calcium, L-type calcium currents, and thereby increased action potential duration. In the cardiac organoids that we develop at Greenstone Biosciences, you can see also functional validation, where atrial cardiac organoids have a much faster spontaneous beat rate and shorter calcium transient duration compared to ventricular specific cardiac organoids. In the quantification, you can see atrial cardiac organoids have a much faster beat rate as well as a much shorter duration of the calcium transient. These are some videos of cardiac organoids generated here at Greenstone Biosciences. You can see from the movie that we can generate large batches of cardiac organoids with relatively uniform diameter and shape of the cardiac organoids. On the left, you can see spontaneous contractions of those organoids. And on the right, you can see the organoids also can be utilized for electrophysiology screening. In this example, monitoring the intracellular calcium flux of each organoid. And this can be used for testing response to drugs and the function and the contractile function of the organoids. And we also can perform omic type of research looking at transcriptomics and proteomics. So here you can see at the bottom recording of cardiac organoids electrophysiological function, and we can relate how that corresponds to the EKG of patients. We can provide these organoids as cryopreserved, ready-to-use frozen cardiac organoids that can be distributed to laboratories or utilized here in our labs at Greenstone Biosciences. So this is a review of some of our products for cardiovascular NAMs that can be these are differentiated cell types from iPSCs. We can generate cardiomyocytes, fibroblasts, and endothelial cells and mix those together at different ratios to either provide pure cardiomyocyte substrates and functional assays, or we can also mix in fibroblasts to try to model fibrosis in a dish for another type of disease model. And we can turn these cardiomyocytes and other cell types into three-dimensional organoids with chamber specificity. We have complex models and panels of iPSCs that can be utilized. We have a diversity panel that can be used for personalized research. The number of iPSC lines that we have are amenable to development of clinical trials in a dish type of approaches to determine the effect of a compound on many patient iPSC cardiomyocytes in vitro. And these cells can be provided as cryopreserved cells, or we can perform toxicity assays here at Greenstone. We also have customized bespoke services where we can perform electrophysiology screening. And always, if you don't see your cardiac NAM or other cell type that you want or disease model of interest, we can work with you to build that model. Our services are always expanding. One expansion is into the generation of gene-edited iPSC lines and other cell types that can be done as a custom service to create the cell types that are needed for your project. We have state-of-the-art equipment and facilities at Greenstone Biosciences. We can provide CRO services. And in this type of paradigm, you can partner with us to send us your gene therapy, small molecule, or antibody therapy, and we can test those therapies for cardiotoxicity using the iPSC NAMs that I have reviewed today that are part of our product portfolio here at Greenstone Biosciences. This is a snapshot of our biobank team. We're a team of highly skilled and trained scientists who are expert in iPSC expansion and differentiation to cardiovascular cell types, as well as many other cell types that are useful for preclinical toxicity screening. So this concludes the webinar. I just want to put our contact information again and encourage you to contact us if you see a cardiovascular application that Greenstone might be able to provide for your drug discovery project or your biomedical research. Please contact us. You can email us or check out our website where we have a request form that can be filled out, and we'll get back to you, and we'll be very eager and excited to work with you and help promote the successful completion of your drug discovery or drug development projects. And with that, if there are any questions, there's a Q&A window. Please put any questions into the Q&A and we will answer those questions. Thank you again for your attention, and we'll go to the Q&A now. So there's one question. The question is, does your voltage and calcium imaging approach catch QT interval alterations and EADs? The answer is yes. The approaches that we use do catch QT interval alterations and also EADs. We can see action potential duration prolongation in response to well-known hERG channel blockers. And also we can see the development of early after depolarizations in response to those same hERG channel blockers. The accuracy is very good from the international validation efforts that have been done by the Health and Environmental Sciences Institute Committee, as well as the FDA, have shown very good accuracy of the iPSC-Cardiomyocyte new approach methods and their predictive nature to predict drugs that will and are known to cause QT prolongation and action potential prolongation. Thank you for that question. Another question is, can left and right ventricular features be distinguished in cardiac organoids? I'm going to say not at the moment. At the moment, the biggest features and the differences are between atrial and ventricular chambers. I know there's work being done in the field to try and recapitulate very chamber specific, like left ventricle versus right ventricle features that are trying to be recapitulated in iPSC cardiomyocytes. Here's another question. The question is how the beating is spontaneous when we are pacing the cells electrically and mechanical stimuli. Could you clarify how they are mechanically stimuli, stretching, or stiffness? So in the approach method that I showed, the cardiomyocyte monolayers are cultured on a plastic dish and they're beating spontaneously. The cardiomyocytes can be electrically paced chronically in culture, and this has been shown in some laboratories to promote the maturation of the cells and to increase the contractile function of the cardiomyocytes. There also are ways to perform mechanical stretching utilizing more viscoelastic matrices to grow the cells on, where the whole monolayer can then be stretched using vacuum to stretch the bottom of the cells. And this has also been shown to modulate the function of the cardiac myocytes and the monolayers. Beyond monolayer type of new approach methods, there are also three-dimensional engineered heart tissue approaches that can be done where the cardiomyocytes and other cell types are actually formed into a muscle bundle or a muscle fiber that can then also be stretched mechanically and also electrically paced. Thank you for that good question. Another question is, how is 3D cardiac organoids differentiation and freeze and thaw protocol about consistency of cardiac maturity? So, in our labs here at Greenstone, we have been doing careful cryopreservation testing to determine the viability and the function of the cardiac organoids following the freeze-thaw process. And we have seen very good consistency and very good matching of the function of the organoids both before and after the freezing process. Here's another question is great data in your slide. Thank you for the compliment. In your slide using the CARTOX instrument, you mentioned the use of a homogeneous culture of cardiomyocytes to integrate effects of drug addition. Wondering if you did the same with added other cell types like endothelial, mural, etc., and did that change the effect of drug treatment? That is a great question. In the international validation study, the hERG committee and the FDA have validated just purified cardiac myocytes. There hasn't been a validation done yet utilizing more complex co-culture approaches. However, it is likely that the introduction of other cell types, which may be more sensitive to a drug's effects, can also promote the development of arrhythmias and can also promote cardiotoxicity. So that's a very insightful question and likely will be the focus of a future validation effort and validation study led by the hERG committee to really be able to determine the effect of other cell types on the drug responsiveness of iPSC cardiac monolayers and new approach methods. Thank you for that question. What is the standard timeline for your cardiotoxicity assays? And does this differ between iPSC-CM monolayers and organoids? So the standard timeline for the cardiotoxicity assay from the time of thawing cells to completing the EP assay is about a 2-week period. And then after about a day or two of analysis, we have the reports completed and can turn around and determine the effect of a compounds on the rhythm of the iPSC cardiac myocytes. The timing isn't too much different between monolayers and organoids at the moment. We can turn around that data at about the same rates between organoids and monolayer assays. Thank you for that question. One more question is, does the beating of cardiac organoids correspond to the physiological heartbeat of the human heart? So for atrial cardiomyocytes, the beat rate is typically around 60 to 90 beats per minute, which is about the range of the normal human heart rate. On the other hand, the ventricular organoids and ventricular monolayers beat at a much slower rate because they don't have as many of the pacemaking channels as the atrial cardiomyocytes. And so the ventricular myocyte and organoid beat rate is much slower, more like 30, I would say more like 30 to 50 beats per minute, as one might expect for the differences in the ion channel makeup and the structure between atria and ventricular cardiac myocytes and cardiac tissues. Thank you for that question about the organoids and the heart rates. Another question came in that says, I'm really interested in using some of your iPSC lines. Thank you. We hope we can get those lines for your use. I've tried contacting through the normal email and have not heard back. So the best way to get into contact with the biobank is to go through our website. in our website, there's a request form, and that request form goes through to our biobank, and we will be able to answer your question and your request there through the request form on our website. And it looks like that was the last question. I'll just wait a moment to see if any more questions come through. And as I'm waiting, I just want to thank all of those who have attended for attending our second webinar. In case you missed any of the webinar or would like to review some points of the webinar and our iPSC cardiovascular new approach methods, we'll make the webinar recording available through our YouTube channel, and also we will post it on our website so that you can review the webinar at your own pace. So I don't see any more questions coming through. So thank you again for your attention. Thank you for your time. I hope to hear from you, and I hope that we can mobilize our iPSC Biobank here at Greenstone Biosciences to help advance your drug discovery project and your drug development projects. Thank you very much, and have a great day.